While we're doing those differential gene expression studies, we see that we apply some treatment and we calculate the gene expressions, we get some numbers, we come up with a distribution, we give another treatment, and then we come up with some numbers, and those numbers are put together in a distribution. So we are comparing two different distributions with one another. So in this way, these kind of tests are two sample tests. So these differential gene expression studies, they involve expression across two or more treatments, and most of these statistical models, they assume that those observations are independent from one another. So these are kind of two separate distributions. So in this way, if you have two separate distributions, we can call it as an unpaired t-test. So where we have two group of samples, where we have one per condition. And our question is, we want to test whether the difference between these two distributions is significantly different from 0 or it is not different from 0. So here we can elaborate that with this picture. So we have, for example, here we might have some treatment and we come up with some individual data points. There may be replicates within this experiment. So we can draw these distribution and we can come up with their mean. Here is the second distribution, maybe a control. We can come up with their means. And then we want to compare these two means with one another. So in this way, if you have two means and we take these two distributions as independent and we come up with their means and standard deviations and we perform a t-test, we call it as a two sample t-test or unpaired t-test or two independent samples t-tests. We can come up with some statistics. So for example, y bar 1 is the average of treatment 1, y bar 2 the mean or average of treatment 2, and same way we have their standard deviations as S1 and S2. We can come up with their standard error of the difference of these two means like this. We can come up with their t calculated with these formulas. So here we put up their differences between their two means minus mu naught. So that is where we have some standard. We might, most of the cases we have 0 over here, but sometimes we are given a situation where we are testing that whether this difference is different than some given number or not. We can have standard error of the means and the difference of the means down there in the equation. Here is an example. So we have the expression of TP53 gene family in patients treated with zinc chloride versus normal persons. So remember, these are all different peoples or different individuals. So we might not find any strong correlations. So we can treat them as two independent samples. So we have 5 numbers in each of them. So our N1 or NA is 5 and NB is also 5. Remember, these are independent or uncorrelated samples. So we run this t-test for two independent samples or unpaired t-test. Here we run it. First of all, what we are testing that the difference is some greater than 0. In fact, we are interested in finding the expression of TP53 in the treated individuals with this zinc chloride is greater than the ones which are the control ones or which are the non-treated. So we want to have some higher means in the treated ones. Actually, we are testing for a mean difference which is greater than 0. So it's kind of a right tail test. Our null is which is negation of alternative. So we have the difference less than equal to 0. We can calculate the mean difference as 5. We can calculate the standard error of the mean difference as 7.5. We plug in into that equation. We come up with t calculated is equal to 0.66. The null distribution has a degrees of freedom is equal to 8. So we want to find this difference on the null distribution, whether it's present in the right extreme tail of this null distribution. In that case, it will be significant. So we draw this null distribution for this degrees of freedom. So over there we have 4 degrees of freedom for this first sample and 4 for the second. So total degrees of freedom we have in this case, in this independent sample test, we have 8 degrees of freedom in total. So we draw the null distribution which is centered at 0. No difference. Let's see where our difference lies. So difference we calculated is 5. The t calculated we have is 0.66. What should be the calculated t, or what should be the critical value of the T if you want to be in upper or top 5%. So in an observation to be in top 5%, you should have the difference between these two numbers should be 13.8, or your T value should be greater than 1.85. Up till that point, 95% values are below and 5% are above. Where is your observation lies? 0.66 lies here, right here. So where your 74% values are below and 26% values are above that. Your T calculated is in fact less than T critical. Same way, your probability of this T calculated is 0.26, which is obviously greater than your critical P value, which is we decided to be 0.05. Since our P value, which is calculated over here, is 0.26, the observation is in upper 26%. It's not in upper 5%. And the P value is not less than alpha or not less than 0.05, we can say this difference between this treatment and control is non-significant. So we see that differential gene expression studies, they involve comparison of the gene expression across 2 or more treatments, and most of the time these treatments are independent of one another.